All right, Mr. Martin's class. Ready for a deep dive into the world of neural signaling. We're going to figure out how messages zip around your nervous system so fast. It's the electrical magic that lets you react, think, even ace those IB biology exams.
It all starts with these tiny messengers called neurons. Imagine a huge network all throughout your body, sending and receiving information constantly. It's pretty fascinating.
Okay, we've got neurons, but how do they actually carry messages? It's like our body has its own little telegram system going on.
Well, instead of dots and dashes, it's electrical and chemical signals. Picture a neuron like a relay runner. Gets a signal at its dendrites. Those are the branch-like structures that pick up messages from other neurons.
So the dendrites are the listeners? Then what? How does a neuron pass the message along?
That's where the axon comes in. It's kind of like a cable, transmitting the signal away from the cell body. Think of it like the runner taking off with the baton.
Wait, if the axon is like a cable, wouldn't that mean the signal just travels passively? But we react so quickly to things.
You've hit a key point. The signal doesn't just flow through. It's actively propagated through a process called an action potential. It's like a series of controlled explosions happening along the axon to boost the signal along.
Controlled explosions. That sounds intense. Is that what those specky graphs we see in class are all about?
Exactly. Those oscilloscope traces are visualizing those action potentials, those rapid changes in electrical charge that travel down the axon.
Okay, so we've got dendrites receiving, axons transmitting, action potential boosting along the way. What about the space between neurons? There's a gap, right? How does the message jump across?
Great question. That gap is called a synapse, and here's where things get really interesting. Instead of electrical, it switches to chemical signals at the synapse.
Yeah.
Think of it like the runner reaching the next checkpoint and passing off a coded message.
So the electrical signal is converted into a chemical message. What kind of message are we talking about here?
We're talking neurotransmitters. They're special molecules released from the axon terminal of the first neuron. We call that the presynaptic neuron. Then they bind to receptors on the dendrites of the next neuron, the postsynaptic neuron.
Like a lock and key. Neurotransmitter being the key that unlocks a specific response in the next neuron.
Perfect analogy. And here's where the nervous system gets really complex. Many different types of neurotransmitters, each with specific effects on the postsynaptic neuron.
Wait, so it's not just one signal. There are different types of messages being sent.
Absolutely. Some neurotransmitters are excitatory, meaning they make the next neuron more likely to fire its own action potential, pass the message along. Others are inhibitory, meaning they make the next neuron less likely to fire.
So it's like a constant balancing act, with different neurotransmitters influencing whether a neuron will fire or not.
Precisely. A single neuron can receive input from thousands of other neurons, each releasing different neurotransmitters. Like a busy town square, all sorts of messages flying around. Each neuron has to figure out what to do next based on all this information.
Mr. Martin's class, hope you're taking notes. Understanding this is key to the bigger picture of how our nervous system works.
Now, before we move on, let's go back to resting potential. Really important to understand this before diving into the action potential. Remember, neurons aren't always firing. They have a stable state when not transmitting a signal.
So it's like the neuron is charging up, getting ready for the next message.
That's a good way to think about it. This resting state, called resting potential, is maintained by a balance of ions inside and outside the neuron-charged particles like sodium and potassium.
Like from chemistry. They're electrolytes, right?
You got it. And these electrolytes are key in setting up the electrical potential across the neuron's membrane.
Okay, this is getting a bit complex, but I think I see how these ions and the resting potential are setting up the action potential.
Exactly. Think of the resting potential as the baseline, the calm before the electrical storm. Setting up the conditions for that rapid change in charge, that is the action potential.
Mr. Martin's class, make sure to review your notes on ion channels and the sodium-potassium pump. They're key players in this whole process.
Now, are you ready to really dig into the action potential itself? We're going to unpack those different phases and see how ions flow in and out, creating that electrical impulse.
Let's do it. This is what I've been waiting for.
Back again, Mr. Martin's class. Before the break, we were looking at resting potential, that baseline electrical charge of a neuron.
Right, like the neuron's just hanging out, ready for a message. What happens when that message arrives? How does resting potential turn into an action potential?
That's the exciting part. Imagine this. The neuron gets a signal at its dendrites, and it causes a tiny change in the membrane potential. If this change is strong enough, reaches a certain level of threshold, it triggers the action potential. That's the electrical impulse that'll travel down the axon.
Like flipping a switch. Once it hits that level, boom, action potential.
Exactly, like a domino effect. The first domino falls, the rest follow chain reaction. And with the action potential, those dominoes are voltage-gated ion channels.
Okay, remind me, what are those ion channels again?
They're like tiny gates in the neuron's membrane controlling how ions flow in and out of the cell. The voltage-gated part means they open and close in response to changes in membrane potential.
So the change in voltage itself triggers those channels to open and close.
Precisely. This is where the sodium and potassium ions we talked about come in. Remember, at resting potential, more sodium outside the neuron, more potassium inside.
Right, and the sodium-potassium pump keeps that difference in concentration.
Now, when that excitatory signal comes in and hits the threshold, the voltage-gated sodium channels open.
So sodium rushes in.
Exactly. Sodium ions, driven by their concentration gradient and the electrical attraction to the negative inside of the neuron, flood into the cell. This influx of positive charge makes the membrane potential rapidly become more positive. That's depolarization.
So the inside of the neuron is becoming less negative, getting closer to zero.
Right. And remember those spiky graphs. Depolarization is that upward spike shows the rapid change in membrane potential.
Okay, sodium in, depolarization happens. What next? Does the charge just keep going up?
Not quite. It hits a peak, then those voltage-gated sodium channels close. But almost at the same time, another set of channels opens up the voltage-gated potassium channels.
Now it's potassium's turn.
Exactly. Potassium's more concentrated inside now, so it starts flowing out. This outflow of positive charge makes the membrane potential drop back down towards its negative resting state. That's repolarization.
So the neuron's resetting itself, getting ready to fire again.
Precisely. But those potassium channels are a bit slow to close, stay open a bit too long, makes the membrane potential dip even more negative than resting potential for a moment. That's hyperpolarization.
Like an overshoot, swinging a pendulum past center.
Great analogy. This little bit of hyperpolarization makes sure the neuron can't fire another action potential right away, ensures the signal travels one way down the axon.
So it's a refractory period. The neuron needs a second to catch its breath before firing again.
Exactly. Eventually, those potassium channels close, the sodium-potassium pump gets back to work, restores the ion gradients, and the neuron's back to its resting potential, ready for the next signal.
This whole process is like a perfectly choreographed dance, ions in and out, controlled by those voltage-gated channels. Amazing how much is happening in such a tiny space, so fast.
It really shows how complex and efficient our nervous system is. Remember we talked about myelination, speeding up signal transmission?
Right, that fatty sheath around the axon, like insulation. Does that play a role in the action potential too?
Absolutely. In myelinated axons, the action potential doesn't travel along the entire axon. It jumps between the gaps in the myelin, the nodes of Ranvier.
Like taking the express lane on the neural highway.
And those nodes are jam-packed with voltage-gated sodium and potassium channels, so the action potential can be regenerated at each node, keeping the signal strong as it travels.
Myelination is faster and more efficient.
Precisely. The diameter of the axon matters too. Larger axons have less resistance to ion flow. Action potential travels faster.
Like a wider pipe, more water flowing through.
Another great analogy. Mr. Martin's class, think back to those oscilloscope traces. Can you see how those different phases of the action, potential depolarization, repolarization, hyperpolarization, show up in that spiky graph?
This is why it's so important to review your notes, make those connections what we're discussing, and the visuals in class really helps you understand it.
We've talked about how the signal travels within a neuron, but the real magic's at the synapse, where neurons talk to each other.
Right, where the neurotransmitters come in, converting the electrical signal into a chemical one.
And here's where the diversity of neural communication really comes in. Different neurotransmitters, different effects, shapes the flow of information through the whole system.
This is where I get a bit lost. So many neurotransmitters all doing different things.
I know, it can feel overwhelming, but don't worry, we'll break it down. Ready to explore the world of neurotransmitters and how they influence everything from our mood to our movements.
Let's do it. Welcome back, Mr. Martin's class. We're still in the intricate world of neural signaling, and now we're at this fascinating place, neurotransmitters.
You know, if we think of neurons as messengers, neurotransmitters are the messages themselves. They're those chemical couriers bridging the gap between neurons at the synapse.
So let's unpack some of these messages. Where should we start?
Maybe the most talked about neurotransmitter is dopamine. You've probably heard it called the reward chemical. and there's a good reason for that.
Yeah, makes us feel good, right?
Well, that's part of it. Dopamine is definitely released when we have pleasurable experiences, like achieving a goal or enjoying a good meal. It kind of reinforces those behaviors, makes us want to do them again.
Like our brain's way of saying, do that again.
Exactly.
Yeah.
But dopamine does a lot more than just pleasure. It's really involved in motivation, driving us to seek out rewards and do things that are good for our survival and well-being.
That makes sense. It's not just about the good feeling now, it's about being motivated for the future.
And that motivation is super tied to learning. When we experience something positive, that dopamine surge helps strengthen the neural connections associated with that experience, makes us more likely to repeat those actions.
So dopamine helps us learn what's good for us and keeps us doing it.
Precisely. But here's where things get really interesting. This whole reward system can be kind of hijacked. That's what happens in addiction.
Oh, right. I remember Mr. Martin talking about how drugs like cocaine affect dopamine.
Those drugs flood the brain with dopamine, creating this intense artificial sense of euphoria. This overwhelms the normal reward system, and then it's harder to feel pleasure from everyday activities.
So the brain gets used to these huge dopamine hits, and then normal stuff doesn't seem as rewarding anymore.
That's a big reason why addiction is so hard to overcome. It's not just willpower. It's about rewiring those neural pathways, getting that healthy balance back in the dopamine system.
This is really making me think about how fragile this whole system is.
It really is. Now, let's shift gears a bit. Talk about serotonin. This one is often linked to mood regulation, sleep, appetite. Low levels of serotonin have been connected to depression and anxiety.
So if someone's feeling depressed, could it be because their serotonin levels are off?
It's definitely a factor. A lot of antidepressants work by increasing serotonin in the brain. That can help with symptoms.
Like the medications are restoring the brain's chemical balance.
That's a good way to put it. Speaking of balance, let's not forget about GABA, gamma-aminobutyric acid. It's the main inhibitory neurotransmitter in the brain.
Inhibitory, so it's the opposite of dopamine and serotonin?
Sort of, yeah. Excitatory neurotransmitters like dopamine and serotonin increase the chance a neuron will fire. GABA's like a brake. Calms things down, reduces how excitable the neurons are.
Like GABA's saying, all right, everyone, chill out.
Exactly. GABA helps us relax, reduces anxiety. Medications like benzodiazepines boost GABA activity. That's why they're often prescribed for anxiety and insomnia.
Okay, so dopamine for motivation and reward, serotonin for mood and sleep, GABA for chilling out. What about those fight-or-flight situations? What gives us that adrenaline rush?
That would be epinephrine, also known as adrenaline. It's released by the adrenal glands when we're stressed, gets the body ready for action. Epinephrine increases heart rate, blood pressure, blood sugar, gives you that burst of energy to face a challenge or run away from danger.
Like our body's alarm system, kicking in when we need it most.
Precisely. And we can't forget acetylcholine, which we talked about with muscle contraction. It's released at neuromuscular junctions, tells muscle fibers to contract.
So every time we move, it's acetylcholine sending that message from brain to muscle.
Exactly. But acetylcholine does more than just movement. It's also crucial for learning and memory.
Wow, seems like acetylcholine does a little bit of everything.
It's pretty versatile. Sadly, its decline is linked to Alzheimer's disease. That affects memory and cognitive function.
So many different neurotransmitters, each with its own job. It's like this intricate symphony, all these chemical signals controlling our thoughts, feelings, actions.
And remember, they don't work alone. They interact with each other, affect each other's levels and effects. It's complex, dynamic, and we're just starting to figure it all out.
This deep dive has really shown me how amazingly complex our brains are.
Well, Mr. Martin's class, I hope exploring neural signaling with you has made you more curious, helped you understand this amazing system better. Remember, this is just the beginning. There's so much more to learn and discover.
Make sure to go over your notes, dig into your textbook, ask Mr. Martin any questions you have. And for everyone listening, if you enjoyed this deep dive into the brain, be sure to like, subscribe, and leave a comment below. We'd love to hear what you think and get your ideas for future episodes.
Until next time, keep those neurons firing and those synapses connecting.